Good afternoon, and welcome to the Department of Medicine's Grand Rounds. Please take a moment to silence your pagers and mobile devices. Next week's Grand Rounds, titled Prediction Models of Chronic Kidney Disease Progression, will be presented by Dr. Shoshana Weiner, Assistant Professor in the Division of General Internal Medicine. It is my pleasure to introduce today's Grand Round speaker, Dr. Antony Rosen. Dr. Rosen is the Mary Betty Stevens Professor of Medicine, Pathology and Cell Biology, Director of the Division of Rheumatology, Vice Dean for Research at the Johns Hopkins School of Medicine, and the Director of inHealth, the Johns Hopkins Program in Precision Medicine and Individualized Health. Dr. Rosen received his medical degree from the University of Cape Town in South Africa in 1984. After completing his internship in medicine and surgery, he pursued postdoctoral studies in immunology at the Rockefeller University in New York from 1987 to 1990. Drawn by a desire for a deeper understanding of human inflammatory diseases, he returned to clinical medicine and was an Osler resident and rheumatology fellow at Johns Hopkins Hospital from 1990 to 1994. He subsequently joined the faculty at Johns Hopkins in 1995 and rose to the rank of professor in 2002. He has been director of the Division of Rheumatology since then, overseeing substantial expansion of the division. His research focuses on autoimmunity in the rheumatic diseases, using subgroups of patients with distinct phenotypes, trajectories, and specific autoimmune responses to define molecular mechanisms. Please join me in welcoming Dr. Rosen to the podium, and Dr. Davis is going to give another brief announcement.
So let me quickly say thank you, Dr. Rosen. We're really looking forward to your presentation. So much so, we have something new. We have a quick survey, if you wouldn't mind filling this out at the end of the lecture. And if anyone comes in late, no one comes in late to Medicine Grand Rounds, could you whisper and ask them to fill out the survey as well? So without further ado.
Oops.
I'll just use this one. Okay, so thank you very much for the invitation. It's very good to be here today. I'm going to talk about precision medicine, and in order to kind of tell you how we got here, I thought I would start by telling you a little bit about rheumatology and some of the specific diseases that I think are being actively answered by these precision medicine approaches, and then I'm going to generalize and talk about precision medicine done enterprise-wide and what the power and what the opportunities are. And I call it wonder and value at scale, and I think it will become clear over time why I call it that. I have no disclosure specific to this presentation. I thought I would start with this quote from Charles Darwin, which he wrote on board the Beagle in the early 1830s. Darwin was cruising the Galapagos Islands and noticed that different islands had species with different modifications. So the finches on one island were not exactly the same as the finches on another. The turtles on one island were not the same as the turtles on another. And he wrote, the islands possess their own species, these species having the same general habits, occupying analogous situations, and obviously filling the same place in the natural economy of this archipelago that strikes me with wonder. Darwin was drawn to this idea that different islands had different species, and he could see the islands looked somewhat different in terms of their microenvironment, and he intuited that there was this powerful biological force that was shaping the evolution of species. Now, Darwin was smart. Darwin also had a lot of time on his side, but he was also incredibly lucky because the spatial separation of the islands neutralized the effects of eons of time. So he was able to, at one moment in time, look left and look right and see millennia in front of him, and he could intuit that there were changes that had been time dependent. And so clinicians are basically use very much the same tools. right? We are observers. Darwin had pretty primitive tools. I'm going to argue that, and used lots of intuition. I'm going to argue that clinicians used to have pretty primitive tools, but have lots of intuition. And, Osler wrote, to make perfect the most difficult of all the arts, the art of observation. And for anybody to say that that art of observation is irrelevant in clinical practice, you're wrong, right? Because it is the basis of most important discoveries. To call to aid the science of experimentation, to cultivate the reasoning faculty so as to know true from false, these are our methods. That was written by Osler in the late 1800s. And I will point out that the power of observation remains unchanged. The tools of experimentation are completely different. I'm going to show you some examples of that. And the reasoning faculty has been massively expanded by the sophistication and the power of modern computation. And so we are in a new phase where the world is going to be reimagined in terms of understanding what causes a human disease and how they should be treated. I'm going to spend a little time on the autoimmune rheumatic diseases because I know them best. And I think that they are so complicated that when I started out as a rheumatology fellow 25 years ago, it was quite clear that we were not going to understand them in my lifetime. And I would say that I've been as surprised as anybody. I believe we already do understand significant parts of the rheumatic diseases, and that within the next 10 years, we are going to understand them in great depth. So the rheumatic diseases are complex in every dimension that you can imagine. They're genetically heterogeneous. The population that have them is genetically heterogeneous. The spectrum of disease is quite diverse. The phenotypes are complex and nuanced. And so rheumatologists get better and better and better the older they get. The more patients they see, the more experience they have. They see subtleties of patterns that they haven't seen before. The phenotypes are not pure, and that there are significant overlap between different diseases. There have been numerous potential environmental forces that have been associated with either the initiation of disease or with flares of disease. And then the diseases are kinetically complex. That is, that initiation of disease and propagation of disease are separated in time. So if you study a military cohort of patients who all have lupus now, and you go back 10 to 15 years and you say, when did autoantibodies first begin? They begin 5 to 15 years before the patient has their first symptom. So these diseases have a long time frame for development. They have discrete events along the time, but nevertheless, they're complicated in terms of kinetics. Once the patient has the disease, the patient isn't the same at all time. They have worsening and they can get better. They can have a disease which evolves over time. So for all these reasons, the rheumatic diseases are exceptionally complicated. The response to exceptional complexity, and this is important to precision medicine, the response to exceptional complexity is one of two things. Study something else, or quit whining and get on with the problem. And I will argue that the tools that humans have at their disposal finally prevent us to actually answer some of these processes. So for the non-rheumatologists in the room, phenotypes matter. The first time I met Mark Hochberg, he was actually over at Hopkins still, and Mark is a consummate clinician, right? Subtleties matter. And so this is the subtleties matter slide. Patterns of disease, looking at simple things at the bedside, the hands, the skin, the kind of rash, what the associations are, tell you a lot about the diseases. And I'm not going to go into anything other than to say on the left side are pictures of patients with dermatomyositis. On the right side, patients with scleroderma. On the top row, no? Yeah. On the top row, this is a patient with diffuse form of scleroderma. On the bottom row, a patient with the limited form of scleroderma associated with anticentromere antibodies, associated with antitopoisomerase antibodies. This column over here is a patient with anti-MI2 antibodies. This is a patient with dermatomyositis and the synthetase syndrome with antibodies against amino-acyl tRNA synthetases and interstitial lung disease. So with very simple but astute observation, you can actually make really important observations right at the bedside. Now, Darwin was lucky because time was neutralized for him by spatial separation of the islands. We don't have such a luxury, right? We don't have such a luxury that we can get people separated by time. And therefore, we have to take the tools that are available to see time's effect. And we have many of them. And so I'm going to tell you about 3 approaches to neutralize time. These are exceptionally powerful. The first one is the concept of memory. I'm going to come back to that. Some event happens, and it leaves a mark, and then you follow it for all time, and that event of the initiating event has memory. The second is that if you're interested in time, you cannot do it in a cross-sectional way. You cannot follow a group of patients at one point in time and say, I can get any insight into time. You have to follow patients longitudinally, and longitudinal data identifies disease trajectories in exceptional ways. The third is that concurrent events neutralize time. Okay, so I'm going to give you those three examples. The first one is memory. Memory, there are many things that lead to memory. I'm going to talk about one, which is immune memory, but there are many other things. There is the epigenome, right, marks and remembers events that last a long time, right? And so there are many tools for computing memory that you have at your disposal. But let's talk about immune memory. The thing that struck me as a rheumatology fellow was these are exceptionally complicated diseases. But in spite of the fact that they're so complicated, the immune system is not at all confused. The immune system knows that if you target lupus, if you target nucleosomes and splicing ribonucleoproteins, it happens in lupus, tightly associated. If you target topoisomerase 1, you have scleroderma, tightly associated. Not just scleroderma, but the diffuse form of scleroderma with interstitial lung disease. If you target components of the centromere, you have a limited form of scleroderma with ischemic digit loss. If you target the amino-acyl tRNA synthetases, you have a form of myositis with Raynaud's phenomenon, arthritis, and interstitial lung disease, right? These are markers of events that occurred which saw those antigens and then drive those immune responses for all time. They captured some event at disease initiation. So memory. If you looked at the antigens that I showed you on the previous slide, they are frustratingly diverse. Lots of different kinds of molecules, subcellular locations, really nothing that unifies them except for the fact that they're targets of the adaptive immune response. And the adaptive immune response, normally if I would just say adaptive immune response, the audience dies. And so I don't go any further than that, other than to say it's very complicated in detail, very simple in principle. The adaptive immune response is turned on when it sees structure that the immune system hasn't seen before, right? And it occurs in a context which is seen by the host as dangerous, which for all intents and purposes is infection or cancer. And if you make that primary immune response, you have this ability to remember that immune response for all time. Okay, I want you to put that to the side of your head. We are saying we have one way to neutralize time is immune memory. Second way is longitudinal data, which identifies trajectories. So this is just to show you some patients who are followed over time. These are scleroderma patients, and this on the y-axis is normalized FVC, and you see that basically the people who have anti-topoisomerase 1 antibody, their FVC drops over time, and the people who have anti-centromere, their FVC doesn't, and they separate into completely separable groups. So trajectory matters. third is concurrent events. And clinicians are amazingly good at concurrent events. I believe it's one of the things that attracts us to medicine, that we see these little connections, that two things happen in time that exceed our filter for saying, like that shouldn't have happened in that person together, right? And we're really good at that. So let me give you an example. This is an example of patients with autoimmune myopathies, and on the x-axis is the time in blocks of a few years, and on the y-axis is the percentage of patients who have cancer at the same time as the autoimmune disease appears. And what you can see is that if patients with dermatomyositis and polymyositis are going to have cancer, they cluster around the time that the dermatomyositis and polymyositis appear. Cancer and autoimmunity cluster together, right? And that's been known since the late 50s, right? And it's been shown many times in populations and in other studies. That confluence is intriguing. Okay, this is a picture for those of you. Obviously, you know I'm from the South, but I'm not from Baltimore. So I'm originally from South Africa, and these are the foothills of the Drakensberg Mountains. Incredibly beautiful. If there are any hikers amongst you, hiking in the Drakensberg Mountain is really an exceptionally beautiful place, very remote. Okay, you'll see why I'm telling you about that, because here's a picture of tepuis off the north coast of Venezuela. And the tepuis off the north coast of Venezuela are these basically mountains that rise up out of the ocean. They're between 5,000 and 12,000 feet above sea level. They have very sheer sides. They're relatively flat. And we got, when I was a Pew Scholar many years ago, to go for a tour of the tepuis off the north coast of Venezuela. They used to take us somewhere great and then somewhere educational. And so the night before the visit to the tepuis, they got a guide who was from Venezuela, but he'd written the article in National Geographic about the tepuis, and he's reviewing the different species of flora and fauna that you're going to see in these tepuis the next day, and he makes the point that the tepuis of the north coast of Venezuela have unique species of flora and fauna that are shared by only one other place in the entire world, and that is the Drakensberg Mountains of South Africa. Now, I know that many of you, even though there is a stereotype about Americans not knowing geography, I know that you all know, particularly in the current era, if you're an NPR reporter, you all know that the tepuis off the north coast of Venezuela are here, and I'm sorry I did put names on the map. The Drakensberg Mountains of South Africa are here. Now, if you look at these two, you would say, how on earth could you ever have unique species that are only found in those two places? And most of you have already know the conclusion, and the conclusion is because at the beginning of time, they were in the same place, right? And the beginning of time was obviously in the primordial landmass. They were connected, and there were these events, geological events, that caused these two things to separate, and they had already evolved these species, and they continued to develop, okay? So the lesson is that you may be looking at two things that appear far apart. You may be looking at cancer on the one hand, you may be looking at autoimmunity on the other hand, but you might also be able to see the primordial landmass, right? So there may be a group of people with rheumatic diseases who have the two things that occur simultaneously that are and represent this primordial landmass. And that if you're able to capture them and study them, you'll be able to understand what events might have been present at initiation of autoimmunity. And I'm going to tell you a very brief story about that before we go on. The story, as with all my stories, are highly clinical, right? Clinical wisdom is deep. So this is from my colleague Fred Wigley, who sent me this email in September 2007, 12 years ago. And he said, "You're right, case proof." 40-year-old lady, healthy smoker. Some Raynaud's phenomenon starts in April 06. Chest X-ray finds a mass. Biopsy positive for small cell carcinoma, treated with radiation therapy and chemotherapy, the resolution of tumor, moderate radiation fibrosis at site of therapy. Chapter 1, April 06. Chapter 2, April 07. Rapidly progressive skin disease from head to toe, diffuse scleroderma, anti-RNA polymerase III positive, low titer topo. What a case. I have serum and plasma. We might be able to get tissue from open biopsy. I don't know. What else do you think we can do? And now we go to the top and we see that don't only say wonder in 2019. I have seen wonder for years. Right? Wow. Right? This is the kind of unique confluence that can really prove things. And I'm going to tell you a very short story. I've got a lot of data, but I'm going to show a very short story. And I'm going to show you how the tools of the modern era can take intuition and can convert it into mechanism. Okay, so the specificity of the immune response and the strong association with clinical phenotype and trajectory provide this critical triangulation in time and space and help unravel mechanism. Okay, so let's just talk briefly about cancer and scleroderma. Dermatomyositis and polymyositis, the association of cancer and those diseases, very robust. In scleroderma, very not robust. And it was really unclear. There were case reports that said, yeah, they occur. There were a few case series. There were also these very intriguing case reports, which we don't have time to talk about, where you can have a patient with scleroderma. In their first evaluation, they have a cancer. You remove the cancer and the scleroderma goes away. Okay, so striking, really interesting connections. So Ami Shah, when she was a fellow, started to try and address this question. And so our question was, did we in fact have the primordial landmass for what turns on scleroderma? And the way we did that was to basically look for, let me just show you this. These are 4 categories. These are all scleroderma patients. They all have had cancer. And they all, in order to get into the study, we had to have access to the tumor, and we had to have access to cells from the patient's blood, and we had to have access to the blood for serum. And what we showed was that there's the striking association on the y-axis is the distance between scleroderma diagnosis and cancer, and these different categories are different kinds of antibodies. So these are patients with anti-RNA polymerase III, antibodies with topoisomerase I, antibodies with to centromere, or antibodies whose specificity we didn't know at that stage, but who were not pol or topo or centromere positive. And when we looked at, was there a group of patients in whom the scleroderma and the cancer come together in time, bingo, that's the group. Now, that was exactly the same patient as Fed had found, a patient with RNA polymerase III antibodies who had cancer occurring exactly the same time. So that is the primordial landmass, and how we know it is because it gives us the ability to go and look in the cancer, to look for the state of the antigen. I'll come back to that in a second. It gave us the ability to look at the immune response and ask what part of the immune response does it recognize what was present in that cancer, and how that immune response changes over time. To do this, we teamed up with Bert Vogelstein, Ken Kinsler, and colleagues, and we asked this question, is there a modification in the target of the immune response in these patients' cancers? And I'm going to go fast through these slides because it's all published, and I just want you to get the overall message. And that is that these patients on the top all have antibodies against RNA polymerase III. These patients on the bottom have scleroderma and cancer, but antibodies against other targets. These are short interval cancers. These are generally long interval cancers. And what you can see is that patients who have Pol antibodies have somatic mutations in the gene for the large subunit of RNA polymerase III, and there is also significant loss of heterozygosity at the POLR3A locus, suggesting that the immune response is editing and negatively selecting for the expression of those mutations. I'm not going belabor this. I'm happy to talk about it. I'm just going to leave you with this model. That is that in the process of transformation and mutation, somatic mutation in this major scleroderma autoantigen induces an immune response initially against the mutated version, and that T cells edit cells that express that mutated version. But what we know is that the mutated version is expressed in the cancer but is not expressed in normal cells. And we also showed that that T cell does not cross-react with your normal RNA polymerase III. So what happens is that this process spreads to the wild type. So it starts off by targeting the mutant, it spreads to the wild type. The wild type cells attack your tissues and also actually give you increased effect against the cancer. Okay. That is a series of studies started with clinical observations that were based on looking for things that neutralize time, that use the tools of the era, next generation sequencing of T cell receptors, high throughput antibody discovery, to subgroup the group of people who have this tight association, and then to define mechanism. I will just point out that once we understood the group of people who had a high likelihood of cancer, we also were able to figure out the people who weren't going to get cancer, who make distinct immune responses. So now you have this kind of precision medicine approach where a patient comes in with new onset of scleroderma, and you can measure if you have antibody X, you have a high likelihood of getting cancer. If you have antibody Y, you have a low likelihood of getting cancer, or you'll never manifest the cancer. And therefore, how you manage the patient initially is influenced by really relatively cheap and easily available, will be hopefully easily available tests. Okay. The rheumatology part I'm going to leave. I just want to acknowledge that discovery of mechanism in human disease and discovery of what you do with that is a team sport and requires people with many different skill sets. I will just acknowledge that work was done as a partnership with my wife, and we have been working together for almost 30 years, and Amish Shah, who is really making major strides in understanding cancer and autoimmunity. And all of these people, both clinical people, clinical investigators, cancer geneticists, immunologists, have played critical roles. Okay. So I'm going to shift now to the more general idea of clinicians in the healthcare system and the things that ail us and really are barriers to bring this kind of thing to everybody, right? This is a great democratizing time with new tools, and yet the access to new tools is really very limited to a small group of people. What clinicians bring is empathy and caring. They bring expertise and deep knowledge. They bring a deep intuition. They bring experience, which is really important, and they bring this vast capacity for wonder. What clinicians are limited by is subtle patterns. So you can have seen lots of patients with scleroderma, and you have seen 2,000, right? But 2,000 has not yet captured the world of scleroderma. And the subtlety if you are a regular rheumatologist who's only seen 40, is totally lost on you. Complex patterns, when things are mixed, I'll show you an example of when things are mixed, you have great difficulty seeing it. Each clinician has problems with large numbers of patients. We're human. And disease is not only a biological phenomenon, it is a phenomenon of the soul. So there is only a certain capacity that you can have seeing patients over and over again and actually have an effective therapeutic relationship. So large numbers, we are limited by the fact we just are not machines. We don't see events, unusual events below critical frequency unless they're amazingly different to what we expect. We are terrible. Humans are terrible at integrating trajectory over time. Terrible. Right? We can't remember the patient's slope was that or that. It's hard to know. Like how long ago before did we see them? Our medical record never displays trajectory to us. I can't speak for the University of Maryland, but I will say that clinical care systems are not 100% efficient. And that is a major source of frustration and loss of joy. And humans become tired and bored. But machines don't. And the beauty of this era is that if we can do the things that we do really well and be augmented in the things that we are either not interested in or that we don't do well, we have a dynamite combination to really move our understanding of medicine forward. Okay, so why do I believe we have reasons to be optimistic? The first one is humans have this amazing ability to sense wonder. The second is, as I've shown you in the rheumatic diseases, is true of every disease. There are important clinically relevant subgroups of disease, and those subgroups are being driven by distinct mechanisms. And third, we are living in extraordinary times where humans have been presented with 3 simultaneous technology revolutions. Technology revolution is the revolution of measurement, where previously each analyte that you wanted to measure was somewhat onerous, required a significant amount of material to do, and had a significant expense. But now, massively parallel measurement has decreased the cost, has decreased the time, and provides humans with enormous opportunity for measurement. The measurement that people think of when they think of precision medicine is basically the genome, right? Precision medicine equals the genome. Untrue. Precision medicine equals measurement. And that measurement could be the genome, the epigenome, the metabolome, but much more interesting at the moment in terms of its ability to get traction is what I call the right side of that curve. The right side of the curve is the stuff that touches the patient directly. Social determinants of health, physiologic measures, things that you measure that actually matter to subgrouping patients in a clinician's office or subgrouping them at home. So first revolution is measurement, and we have amazing opportunities to harness that for subgrouping. The second is the increased power and sophistication of analytical approaches and also of data visualization, right? Extraordinary if you think about taking huge amounts of data that no human could analyze and find patterns, right? And do it relatively fast. We don't have the capacity to do that, but computers do. I'll give you some examples of that. And then this last issue is the revolution of connectivity, right? The old form of medicine, every significant interaction between a patient and a provider had to happen face-to-face. The new world, face-to-face is very inefficient. I mean, I'm surprised how long it took me just to get across town. It's maybe 3.5 miles. It really is extraordinary, right? The efficiency gains from increased connectivity are incredible, and we need to take advantage of those. Okay, so I'm going to tell you a little bit about the Hopkins Precision Medicine program, and you'll see that our program is an outward-looking program and we believe has great capacity for interaction. We call it inHealth, based on information and health. And inHealth is using the era's transformational tools of measurement, data science, and connectivity to discover clinically relevant and mechanistically anchored disease subgroups at scale and to deliver what we learn to impact the value of healthcare. I'm going to unpack that definition as we go through, but I want to point out a few things. The first one is that inHealth is not about measurement. Precision medicine is not about measurement. Precision medicine is about patients, right? It's about patients. And therefore, our key need is that what we measure when we define a subgroup matters clinically, right? If it doesn't matter, then at the moment, we are not interested in it. That doesn't mean it's not interesting, and it doesn't mean it won't become interesting. It just means that at the moment, it's not interesting for proving that precision medicine actually has value. The second is this mechanistically anchored. Subgroups are important, But if the subgroups don't lead you into understanding why the subgroup happens, that mechanism could be a genetic mechanism, it could be some other kind of biochemical mechanism, it could be a social mechanism. But if you aren't thinking about mechanism, then you are not going to have the tools and the levers to intervene. And then the third thing is that it's great. We have all been, and I count myself among this group, we love the chase, right? We really love the chase. When it smells new and nobody's been there before, and that's what we go after. And then we find that and we chase the next thing, right? But our responsibility to society is that when we find something, that we deploy it and deliver it and continue to study whether it matters. And so this last thing of delivering and learning so that we can impact the value of healthcare, I think is incredibly important. Okay. The inHealth program is a one university program, which means that we have the School of Medicine is a big part of it, the health system's a part of it. We have a strong collaboration with the Applied Physics Lab on building the platform I'm going to tell you about in a minute. The School of Public Health, which is somewhere over here, down there, is really critical on some aspects. The ICTR, which you are now a partner in, is an important part of this. And so we really think that many different components are going to bring value. This is a group which we call the executive group that basically meets every week. It has people from all parts of the organization, and it often keeps us honest. I'll say somebody, for example, like Jody Siegel, who says so. Like, did such and such a center show that there was value, and you'll see her rubric for defining whether there's value, and if they don't show its value, these people get serious heat. Dwight Raum is the chief of digital strategy for the thing that I'm going to tell about in a minute. We also have the steering committee, which has many people from the ICTR on it, and we meet monthly and really review many of these difficult problems with doing precision medicine. Precision medicine is difficult to do in an easy, efficient, and patient-friendly way. inHealth has three different components. I'm going to tell you about all three. The first one is what we call Precision Medicine Centers of Excellence. The second one is the Precision Medicine Analytics Platform. And the third one is the inHealth Science Core, which is the suite of measurement, analytical deployment, and economic experts and tools which allow us to harness value from subgroups at scale. And I'm going to go through each of those in turn. Centers of Excellence. The first one is all about the patient. Patients are partners in discovery, and these are often multidisciplinary centers. Patients are followed prospectively over time, collecting research-grade data and samples in the context of care. I'll give you an example of that next. For every center of excellence that we've started, there has to be a clear scientific framework, and it has to be focused on clinically relevant and mechanistically anchored questions. There has to be a clear value framework with milestones, and it has to focus on both discovery and delivery. These are the centers that we've started to date. We are starting approximately 6 to 10 new centers a year, and we hope to have more than 50 centers by 2024. What we learned pretty early when we started these centers up was that academics are very good at building their own copy of things. I love it, absolutely love it. The problem is that single copies are very hard to scale. And so the first two that we built, which were prostate cancer and multiple sclerosis, I used to have a foot hell of hair, and it was pitch black, and that was 2.5 years ago. It was a hard lift. I will say it's a very hard lift. And what we realized was that we had to change the model from the beginning. So everybody else got started in a totally different system. And the system now is highly constrained and templated. So what we could learn from the first two, we made this thing which we call Precision Medicine Center of Excellence in a box. And in order to be a Precision Medicine Center of Excellence, the box has a bunch of different modules, 6 or 7 modules. You have to do the work, and you've got to go down. You have to drive down the highway. There are on-ramps, there are off-ramps, but everybody has to use the same highway. And we have found that while there was a little initial resistance, the amount of work that it takes now to start a center is so little that we've lost the resistance. Very important. Precision Medicine Center of Excellence in the box, really, really helpful. Okay, so these are the ones that we started. I'm just going to give you a couple examples. So we learned a bunch of things. Some people came with amazingly good ideas. Multiple sclerosis, Peter Calabrese and Ellen Mowry really worked amazingly well, and they took direction well. That's why I'm not talking about prostate cancer. Who, by the way, are doing great, becoming my favorite children. But what Ellen and Peter did, which is so helpful, is that the most important thing about precision medicine is the data in. The data in, the clinical data in, is unfortunately, every clinician does it according to their own way, and they do it what's efficient. And I don't know about your Epic, but when our Epic was installed, wasn't that user-friendly and didn't really enable people to get any bang out of the adoption. And so we agreed. They came up with this Epic smart form that they wanted to implement across the Center of Excellence. They had never collected prospective data in the MS Center at scale, never done it. Right? They'd done it only as components of Study A or Study B. Right? And they said, we want a smart form, we know exactly what we want in it, and there's certain things that are very onerous and time-consuming for MS clinicians, and we want that problem solved. And that meant that you have to pull a bunch of data in. And in fact, when you start off, you actually have got to get sometimes a human to enter the data as you begin. So there is some upfront investment in getting this started. Once you have people on, the entry with each new patient is really not that great. And we said, okay, great, we'll do this smart form. Within a year, we had data on 3,000 patients, all highly templated. But what was interesting was that wasn't only the MS Center of Excellence clinicians that were using it, everybody in the MS Center was using the smart form. But why was that? Because the benefits to them in routine care was so great that it was a no-brainer for them to say, I'm going to just fill this in because it keeps on populating these critical things about the patient, events, responsiveness to therapy in the past that was so helpful to them. They're currently doing a trial at a smaller regional health system where they've adopted and are implementing this smart form. And clinicians say that it cuts almost 10 minutes off their visit any time a decision needs to be made. That is a huge savings. So templating. Second, using a bunch of clinical phenotyping tools in the waiting room, tablet-driven, that the patient actually comes in and does, that populates the record at the right moment. So there are a bunch of patient-reported outcome measures. There are tools of, for example, related to patient mobility, the walking cadence, the writing sample which is analyzed, that are all computed and brought in to the patient chart by the time the patient sees the clinician. Bringing the right biobanking, bringing retinal imaging to each, so these are optical coherence tomography scans. This other one was a major win. Where you get your MRI can matter, not only because the machine matters, but because the protocol matters. And that is even within the Hopkins system. People were doing different protocols in the Hopkins system. That made no sense. Out of the Hopkins system, the same was true, right? And so they basically agreed on what a standardized protocol would be that was implemented throughout the Hopkins system, and also a large regional radiology player implemented the system for that particular protocol for Hopkins patients. And all the data can flow into what I'm going to show you next, which is this precision medicine platform. Right? These are extraordinary efficiency gains for discovery. Okay. Let's talk about PMAP. So PMAP, let me just tell you what the essence of this thing is. So there may be some IT people and bioinformatics people in the room, and when they see a kind of thing like this, there is nuance that they are going crazy over, and I'll just advise you, don't ask me about those nuances. But I will tell the rest of you what's important. The first thing is that bringing the right grade data onto this data platform is incredibly important. And so Chris Shute has been a very important part of how we ensure that the clinical record that comes on is predictable and is the right kind of data, the right quality data. We bring data from many, many, many different data sources. They're all marked on their way in with metadata related to that patient and that visit. And they go into what Chris Shute has called this data lake. Basically, instead of putting it into very well-arranged tables and stuff, they just go into this big lake, right? And then if you're interested in X patients with Y characteristics, then you can pull them up, and they will come up through this IRB filter, and they will come up into this projection that people can use. The data lake is basically filled with a huge amount of data in a highly safe environment. The data itself never leaves that environment, but projections of that data become available to investigators to use. There are many, many benefits, right? One benefit is the data is in a safe environment. And what that did is it changed dramatically the way the IRB looks at the need to specify which data fields you want in your study and how many records you want to look at. And so normally that can go back and forth with the IRB for a long time, and you say, I forgot I wanted to look at X, and you'd have to add something else. Okay? That is completely gone. This is now a tier 1 risk platform that makes it incredibly easy. And then delivery of that data is in a separate thing which we call the delivery platform that is currently being built that can take insights about subgroups that you learn here and then re-implement it into patient care. I'm going to miss that out. I'll just say the value frameworks are incredibly well defined. I will give you some examples. I'm just going to go through this top one. As you implement your innovation in practice, will you be collecting any of the following outcomes? Measurement of implementation success, measures of improved delivery of care, measures of improved patient experience of care, and basically for every new center they have to go through the structured process. And if they don't, you'll notice that there's a lot of questions here which say, why not? And if they don't, they have to do it again. Okay? Until you come up with a good why not, you have to fill in the box. Okay. Okay, so how will the value of precision medicine be realized? So I think the first thing is, this is designed to occur at the interface where patient care occurs. Precision medicine is about the patient. Therefore, it has to occur at that interface. We're focused on these clinically relevant subgroups. There is this capacity to capture high-quality data. The PMAP allows easy use for discovery. There are lots of measurement and analytical tools that are already embedded close to PMAP or on PMAP. So not every individual has to make up their own, right? There's a suite of things that you can use to say, so-and-so worked for such and such a group, and you can just try. These are pretty easily applicable. And we have designed our system to facilitate interactions and collaborations both internally and externally. And I think the new world is going to require that data is going to want to be shared, and it has to be shared in a highly regulated and transparent in an appropriate way, but it has to be shared. Okay, I'm going to leave that one out. Actually, I'll just say one other thing. I think that there is the issue about the state of Maryland. It's very unique in the challenges and opportunities of the modern era. I think Maryland is suffering more from changes in healthcare reimbursement, but I believe that precision medicine is going to be a major opportunity for us in that regard. I'm happy to take any questions.
Thank you so much. It's wonderful. Questions? Comments?
We're all envious, obviously. I mean, this is a great infrastructure you have created. So who regulates access to data, or is it regulated at all? Can anyone use the data, and who would QA uses of the data on the outside?
Yeah. So currently, there are several uses. Let's just talk about research use first. So research use, everything that currently is used for research is being evaluated by the IRB, and the IRB protocol is the filter to let the data projection occur. So on the research side, that's the way. Our belief is that with time, there probably are going to be other ways to use the data. For example, there are going to be QI projects, which we brought a lot of new data on, which QI people want to use. QI and quality assurance are going to be able to use it without IRB, and there will be a special filter that allows them to use it. I think the financial group also will use data. It doesn't make sense that you'd have to have like 5 different platforms. It's one set of data, if you bring everything together, that there would be a series of ways that they would be allowed to touch them. All of them, it's like got this hard shell. So in order to see it, you have to get the data projected, and each one of those events is going to be regulated.
So Dr. Rosen, so if we wanted to set up PMAP at
Yep.
UNB,
Yep.
you've lived through this, what would be the 3 hints that you would give us, and that the mistakes that you made 2.5 miles away?
Okay, so I think the first thing is that even though it seems very costly to start with, it is actually not that costly. If you multiply the amount of time, energy, effort that each individual group is using to access data, and the amount of time that the IRB is often just the process of going back and forth delays that, there is a significant cost that you're going to mitigate. And I think talking about that, quantifying that, and showing that, I think is a very important part, because people who are spending money want to know that they're going to save money somewhere else. The second, I think, is the kind of the bioethical framework for using PMAP, I think, is very important. And so I would put effort into that at the beginning. I think different institutions currently have different kind of thresholds for that really makes me uncomfortable, and this one doesn't. But we have a very, very high threshold for that, and probably that should come down with time, but I would be very explicit about that. And then I think the third is that I think eventually it would make sense, and particularly if PMAP has already been built, it makes sense not to build it again. It makes sense to use the things that have already been built, and you don't have to mix data. The beauty of the way that this PMAP is built is that it uses federated data, so that you could have an instance of PMAP sitting next to, which is a Hopkins PMAP, which could sit next to a University of Maryland PMAP, which could sit next to some other body's PMAP, and that the data would not mix unless you decide explicitly we would like to collaborate around X or Y, in which case you would explicitly decide to make a mix. And that mixture would be yet another level of projection. So I think that the goal would be to get a large technology company to want to make instances of PMAP that you could get off the shelf, and that they would continue to improve them and watch them over time. That would be the goal, but we aren't there yet.
How critical is your safe desktop?
Very important. Safe desktop is important, but it can be replaced by you notice there was this thing called the Jupyter Notebook.
Yeah.
You don't have to use safe. Jupyter Notebook works just as well, and Jupyter Notebooks are easily accessible. But I do think that having a venue where investigators who are credentialed can, in a safe environment, examine the data and manipulate the data is critical, because otherwise I think it could be the Wild West.
Didn't it?
Yes, thank you for a wonderful talk. I'm wondering if results from clinical genetic testing of animal tests or exome sequencing gets pulled into the data lake.
Yeah. The data lake is completely data agnostic. And the amount of data and the complexity of data, there's huge capacity. That's the beauty of the cloud, is that there is a large amount of place to put it. Unfortunately, it can get expensive, but at the moment people are still competing for cloud business. But I think that is really a major opportunity.
Any other questions, comments? Well, let me thank you again. Wonderful.
No problem. Thank you.
Because there you only see a cross-section that you process theory and partner. And we've done so much analysis of cases where you find a single follow-up or even if you do something but you can actually try to predict what's not happening with plasma. So it's a detail. I didn't want to ask you in public because it's